Hello, everyone. Welcome to the first public outreach lecture of the Hock Tan and Lisa Yang Center for Autism Research at Harvard University. My name is Chin-Fei Chen, and I'm one of the center's principal investigators. We just launched this center about a year ago, thanks to the generosity of Hock Tan and Lisa Yang. Our center focuses on the biological basis of neural development with the goal of better understanding autism spectrum disorders. We are delighted to see everyone gather here today and hope to see some of you in person for our next public program, which will likely be hosted in partnership with our sister center at MIT. I'm delighted today to introduce our speaker, Professor Christopher Walsh. Dr. Walsh's lab is one of the 8 core labs in our center. Dr. Walsh is an investigator of the Howard Hughes Medical Institute. He's also the chief of division of genetics and genomics at Boston Children's Hospital. He is the Bullard Professor of Pediatrics and Neurology at Harvard Medical School and the director of the Allen Discovery Center for Human Brain Evolution. Dr. Walsh is a neurologist whose lab studies genes and mechanisms that regulate the development of the human cerebral cortex. His lab has identified dozens of genes responsible for human developmental brain diseases like epilepsy, autism, intellectual disability, and has found that a few of these genes are targets of the evolutionary process that shapes the human cortex. His talk today is entitled Autism Genetics, Cognition, and Human Brain Evolution. After the talk, if you have any questions, we ask that you raise your hand to ask questions, or if you prefer, you may also type questions into the chat box at any time. So Chris?
Great, thank you very much, Chin-Fei. And I want to thank the Tan Yang Center for Autism Research for giving me the chance to inaugurate what we hope will be a very lively series of outreach seminars. And so I'll be talking today about autism and the genetics of autism predominantly. The talk will be a bit more of a classroom lecture than a typical seminar, given the broad audience that we're trying to reach with these seminars. It's adapted from a lecture I give to the Harvard medical students as part of their genetics course. But I couldn't resist the opportunity to put in a little bit of unpublished data near the end. I'll be talking mostly about not inherited genetic causes of autism spectrum disorders, as well as some inherited causes. Very little about the mechanism of autism genes, because that's a huge lecture all by itself. And at the end, I'll talk a little bit about how we can use human genetic disorders to identify parts of the genome that may have had roles in human brain evolution. So how do we define autism? That's open to some debate, but it's generally recognized as sharing a triad of impaired social interactions, impaired language, and repetitive or stereotypic behaviors, typically diagnosed at the age of 3 or 4, although we're increasingly trying to diagnose it earlier. Originally thought to affect maybe 1 or 2 in 1,000 people, but the broader autism spectrum may now include up to 1% of children, or perhaps even more. There are frequent comorbidities, cognitive impairment in about half of kids. A minority of kids show some regression, and seizures and epilepsy occur in about 1/3. So there's a lot of overlap with other neurodevelopmental disorders that affect things like epilepsy and motor coordination. Now, the term autism is a very difficult term for a scientist because it is not really very crisply defined. It's an observational diagnosis. It's not always clinically stable. Children actually very often move into and out of the diagnosis. In fact, we hope that if we get at them early and give them intensive treatment, they'll move out of the diagnosis. That's not true, however, of a lot of genetic traits. There's no clear correlation with the genetics, as I'll show you. There are probably more than 1,000 genetic causes. There's no clear correlation of the term autism per se with prognosis, because we know that some kids with autism have high function and do very well in school, but they may have awkward social behaviors, whereas other children have difficulty in all developmental spheres and have a much poorer prognosis. There's no clear correlation with pathogenesis. And there is substantial causative and treatment overlap with other conditions. So having said that, we nonetheless use the term widely because doctors as well as parents, we recognize this sort of characteristic triad of social difficulties that falls under the rubric of autism. So there's very strong evidence that genes play a big role in autism. This was known long before we understood what those genes were, and it was based on observational twin studies. And twin studies suggest that autism is one of the most highly heritable neuropsychiatric conditions. And this is learned by comparing the concordance rate of autism among identical twins who have essentially 100% of their DNA shared, and that of non-identical twins who share only about half of their DNA. And so if we do that, we find that dizygotic twins share about 50% of their genes. Monozygotic twins share essentially 100% of their genes. They have similar environmental exposures. If one dizygotic twin carries a diagnosis of autism, there's a 10 or to 30% risk that the other twin will. But if the twins are identical, there's a 70 to 90% concordance risk. And this suggests a very strong role for genetic causes, although no one is trying to argue that genetics is the only answer. And so what sorts of genes then might be involved in autism? So genetic disorders have a variety of contributions. We recognize rare Mendelian syndromes that often have names, things like sickle cell disease or tuberous sclerosis or Fragile X syndrome or Rett syndrome. These are caused by rare mutations, meaning they're very uncommon in the population. And almost everybody who has that particular mutation gets the disease. On the other hand, we hear about common variants that may influence our risk in a very, very combinatorial way. This is particularly common for diseases of old age like high blood pressure and diabetes. So are the autism mutations then rare or are they common? These rare mutations are under strong negative evolutionary selection, meaning that they're bad for you. They make you sick. They decrease the chances that you'll pass those genes on to your children. And so they tend to remain rare in the population. And it turns out that it's rare for kids to get severely ill. And the reason for that is often due to these rare mutations that present themselves right at the beginning of life. Now, among those, just to briefly remind some people about the different patterns of genetic inheritance. Genetic disease can be recessive or dominant, or it can be more complex. And so things like sickle cell disease are single gene recessive disorders. We have 2 copies of pretty much all of our genes, one from the mother, one from the father, except for the X chromosome in males. And recessive diseases, you do just fine if you're a silent carrier with a mutation on one of your 2 copies, but you get the disease if you inherit disabled copies of a gene from both parents at the same time. Single gene dominant conditions, in fact, you need both of your genes working, otherwise you'll get the disease. Examples of that are Huntington's disease, neurofibromatosis, and as you'll see, many forms of autism are single gene dominant. And then there are complex traits where there are many hundreds, maybe thousands of different loci in the genome that all contribute a tiny bit, 1% or 2% to risk, and they act in a combinatorial manner. Turns out there's roles for all of these types of genetic contributors to autism. And finally, before I start talking about autism specifically, I want to point out to people that may not be aware of it that not all genetic traits are inherited. We think of the terms inheritance and genetics as synonymous. However, genetic traits can be inherited, but in fact, they can also appear spontaneously or de novo in a family. And so if we think of this imaginary family with 4 children, roughly half of each child's genetic material comes from the father, roughly half comes from the mother. But in fact, in every generation, there are about 100 mutations that occur in the mother or the father's DNA that are present in the child and absent in the parent. Fortunately, most of these are not functional and have no consequence. Only about 1 of them occurs in the part of the genome that actually codes for protein. But these de novo mutations in our exons I'll show you, are an important cause of autism. So I'll go through more or less the history of where we stand with the genetics of autism and leave you with the sort of areas that are under active research. And some of that research is being very actively followed up with the support, the generous support of the Tan Yang Center. So 15 years ago, we knew very little about the causes of autism, and there was tremendous debate about whether there was even a genetic component to it. And many doctors were firmly in the camp that it really wasn't a genetic condition at all, because it didn't typically run through families. Typically, an affected child would have normal parents. At that time, we knew that there were some single gene disorders like Fragile X syndrome and tuberous sclerosis that could make kids look autistic. But in fact, many people, again, consider that not to be real autism, but to be some other sort of a condition. And then there are also a couple of rare chromosomal disorders that are also associated with autism. So if you think of the genetics of autism as a pie, there were a couple of slices that had been defined, accounting for a couple of % of all kids that came to the clinic. One of the big revolutions that came then about 15 years ago with the development of ways of, thanks to having our whole genome sequence, we are able to then probe our genome very precisely to look for tiny missing segments of the genome called copy number variants or deletions, or segments of the genome that were duplicated, such as here, where segment C of this particular chromosome has been duplicated and inserted in tandem. And the methods for doing that are called chromosome microarray, and this is now a routine clinical procedure and has been for about 15 years, where you take the patient's DNA and reference DNA from the normal population, and you put it on a glass slide or on some sort of substrate that has little probes for many, many, many different loci in the genome. And you compare them. And if you have a green signal, it means that there's a missing piece of DNA in the patient compared to the control. Or if you have a red signal, it means that there's too much of a particular part of the genome in the patient compared to the control. So when this was done with autism, it was done in families where the parents were normal and the child was on the autism spectrum. And it was very clear that a significant minority of kids with autism had de novo copy number variants, where the parents had the normal colors, normal yellow throughout the genome, and the child had a gain or a loss of a segment of the chromosome. And these happened in about 6% of kids with autism, as shown by the left bar, and they could either be a gain or a loss, shown by the blue and the red. And the siblings, the normal siblings, at a much lower rate of these copy number variants. And so this means that most of the copy number variants seen in the autistic kids probably contribute or account for the disease, but maybe not all, because the normal siblings sometimes carry them as well. But if you actually compare what's in the segment that's duplicated or deleted, you can see that here now shown the normal kids had very small copy number variants that were less than 1 million base pairs or so in size, whereas the probands shown in green had larger chunks of the genome that were duplicated or deleted. And in fact, the siblings had relatively few genes involved, whereas the affected kids had large numbers, often dozens of genes duplicated or deleted. And so this suggests that many autism genes look like they're especially sensitive to gene dosage or balance, and suggests that maybe some of them act in this sort of de novo and essentially a dominant way, where if you disrupt or duplicate one of the two copies of a gene, it can create risk for autism. And this suggests that these copy number variants contribute to about perhaps 5% or so of cases with autism. And in fact, when you look across the whole genome, it turns out there are certain parts of the genome that are frequently involved by these copy number variants recurrently. There's the most common one is on chromosome 16p11, where you see a large number of kids in this particular study had deletions or duplications. There's also a recurrent site on chromosome 22, another recurrent site on chromosome 15, and a few others elsewhere in the genome. However, you can see also that copy number variants in lots of different places in the genome can cause the same disorder. And in fact, a kid with a copy number variant on chromosome 2 can look indistinguishable in the clinic from a kid that has a copy number variant on chromosome 16. After we started discovering these, we found that very similar copy number variants contribute not only to autism, but also to other conditions. So the same 16p11 deletion or duplication that can create risk for autism also can create risk for schizophrenia or attention deficit hyperactivity disorder or intellectual disability or epilepsy, or can be found in people that have normal cognition, remarkably enough. Similarly for an orexin 1 deletion or 22q11. And so this is a mystery. Why are there many genes that can all converge on one disorder, where you can get a copy number variant in so many different places in the genome and look indistinguishable in the clinic, or where you get a copy number variant in the same place and one person with that exact change can have autism and another one with the same change can have schizophrenia? And this is a fairly profound mystery that we don't really have good ideas for understanding. So after the understanding that de novo mutations were important, the next technological revolution came with our ability to sequence the genome relatively rapidly and cheaply. And this was initially done just for the parts of the genome that code for proteins known as the exome. And so genes are very long, and they have parts called introns that are cut out and not represented in the protein, and other parts called exons that are actually that actually encode the amino acids that are responsible to put together the protein. And so the next question was whether if de novo copy number variants are important, is there a role for de novo point mutations where a single base pair out of the entire 3 billion of the genome is substituted from one base pair to another? So these are called point mutations. I'll also refer to them as single nucleotide variants. And so these are changes to the genetic sequence that typically affect protein structure. And here's an example. This is the genome uses a 3-letter code made of thymine, adenine, cytosine, and guanine. In this case, a TAC encodes for an amino acid called tyrosine. And so this particular codon can be disrupted by mutations in a variety of ways that can either be silent or can be terribly important. For example, if this C at the end of the codon is changed to a T, that also codes for a tyrosine. And actually, it doesn't change the protein at all. So even though the DNA sequence changes, there's no effect on the protein. And so you would expect this to actually create a normal protein and for the gene to function just fine. Alternatively, you could change the A to something else like a G, and that would swap the tyrosine for a different amino acid called cysteine, and that may or may not be damaging. That's called a missense change, and sometimes changing an amino acid can mean all the world to a protein. It can turn it inside out. Other times, swapping one amino acid for another can have essentially no effect. And so some missense changes are well tolerated by proteins. And finally, you can change this C to a G, and that's really bad news because that encodes a stop codon, and that basically cuts off the protein right there. And the rest of the protein that's still to be made, hundreds more amino acids, is just truncated off. And that creates a protein that's smaller than normal. And those very rarely create functional proteins and are almost always damaging. And so when people started sequencing the exomes of parents and their children with autism, they found that the more damaging the mutation was, the more highly enriched they were in autistic kids compared to the control kids. And so that's seen in this chart over here. And I'll keep using these sorts of bar graphs over and over to compare probands and their siblings for all sorts of different kinds of genetic changes. Here you see that the silent mutations so the overall number of mutations was the same in autistic kids and in their normal siblings. It's like real estate. It's all about location. So sometimes the mutations are silent, and those are equally common, those silent mutations in the unaffected and the affected children. These non-synonymous or missense changes, you can see that they're a little bit more common in the probands and the siblings. And then the most damaging mutations, these nonsense or stop mutations, those are very rare sorts of mutations to happen because there are only certain changes that create such a stop codon, but you see that they're enriched about 4 or 5-fold in the autistic kids compared to their normal siblings. And so this provides strong evidence that as a whole, these coding region mutations are an important cause of autism. But in fact, when you look at the genes that carry these, there are hundreds of different genes. And so we knew that there was a role for them, but it took a little bit longer to figure out exactly what all of the different players were. And so that's where sequencing now thousands of children begins to identify hundreds of genes that are currently mutated in autism. And this is a figure from a paper published just last year in Cell by, as you can see, a very large number of collaborators from all over the country. And the genes that are in bigger print and higher up have extremely strong statistical evidence that they were currently mutated in many kids with autism. These are now familiar players. SCN2, which encodes a sodium channel, SYNGAP1 that encodes a synaptic protein, CHD8 that encodes a chromatin factor, and chromatin factors are a big interest of us in the Tan Yang Center at Harvard. Other ones like FOXP2, another transcription factor, and so on. As you get down below, the genes are mutated less frequently in kids with autism, and the evidence that they're involved gets lower and lower, and there are various statistical thresholds indicated by those dotted red lines. But basically, we've already implicated well over 100 genes that are familiar now autism genes. And that's very useful in the clinic to help us diagnose it. But again, these genes too typically create risk not just for autism, but for broader neurodevelopmental disorders, things like delayed walking or delayed speech, intellectual disability, or epilepsy. Now, when we sequence really large numbers of kids, we start to see genes that are a bit more selective to cause non-autistic traits like motor problems and so on. And that's shown here by the blue dots, where in fact, these genes are a little bit more commonly mutated in kids with other kinds of developmental problems like intellectual disability or delayed walking, and a little less common in autism spectrum disorders. And then these yellow ones are a little bit enriched in autistic kids relative to these other kids with more severe neurodevelopmental disorders. And so there are some genes that look like they might be a little bit selective for autism, some that might be a little bit more selective, but it emphasizes that by and large, there's mainly a lot of overlap between autism and other developmental disorders. And so again, why are there many genes that cause one disorder, as well as one gene that causes many different neurodevelopmental disorders? And again, this, as I say, I pose this as a question, but not something I have an answer for. So what I've told you about so far are autism mutations that are de novo, that are present in a child who's affected and not in the parents. And these de novo mutations is what helped us understand how this paradox that why autism can have a large genetic component and yet not travel through families. And that's because once a child has this mutation, if they go on to have children, they can certainly transmit it to their children. So it is inheritable, but in that particular family, it is not yet inherited. But then how do these de novo mutations account for what I just told you about, this heritability of autism that the identical twin studies tell us about? Well, these de novo mutations really don't easily explain the heritability. So it does suggest that there are some inherited causes. I'll tell you next about recessive mutations that are in fact inherited in families. And a large-scale study in 2019 led actually from Boston Children's Hospital, from our division by Tim Yu and Ryan Doan from my lab, again, sequenced large numbers of kids and showed there's a very important role for recessive mutate genes where both copies of the gene are knocked out. And this contributes to up to about 3% of kids of autism overall. And you can see that the excess of these recessive mutations in cases shown in red compared to their unaffected siblings shown in blue is especially striking in females, where there's about 4 times as many of these recessive gene knockouts in cases compared to controls. And so these recessive mutations seem to be particularly important in females. And actually, we haven't fully explored yet exactly the contribution of recessive mutations, because this study mostly looked at the most severe, those nonsense mutations that I told you about, and actually really has not yet fully absorbed the analysis of these missense mutations that are trickier to analyze. And so I think 3% is probably a lower limit. So just now, a couple of quick words about the function of all of these genes. I've told you that there's more than 100. There's probably more. There are several hundred genes that have been implicated in autism. They do share, by and large, the common theme of regulating the synapse, although they regulate the synapse from many, many different levels. So some genes that are mutated in autism encode actually proteins that actually make up the structure of the synapse, things like neurexin and neuroligin that tie the presynaptic neuron to the postsynaptic neuron, or they make up the channels that regulate the excitability of the neurons, calcium channels, sodium channels, other sorts of channels, or they represent signaling proteins like SHANK3 that are in the postsynaptic protein and that start the signaling that goes into the nucleus. But then they also actually can encode proteins that are in the nucleus that regulate gene transcription, because it turns out that the response of the synapse and the targeting of synaptic activity to memory and to learning involves this whole chain of events that starts from the activity of the synapse, that goes down to the nucleus, that stimulates the expression of new genes that might have been silent before the neuron was stimulated, and that create then new proteins. And so there is a general consensus that these genes implicated in autism regulate the plasticity of the synapses, because the plasticity of the synapses underlies our learning and memory. So that autism is, in that sense, broadly classified as a sort of a disorder of learning. So what I've just then told you about is that we've seen it's been an extremely gratifying change that I've seen over the course of my 15 years at Boston Children's Hospital, where we've gone from, again, arguing about whether autism was even genetic or not, to the point where now if a kid comes into the clinic and we do a chromosome microarray and we do a whole exome sequence, we have about a 20, 30, or 30% chance of getting a specific genetic diagnosis for that family. Now, that leaves a lot of the heritability of autism yet unexplained. We've gotten to maybe a third of the pie chart. And so what is the rest of it? I told you that the heritability of autism, the part of autism that we expect to be ultimately explicable in the form of genetics, is something like 60 to 90, as high as 90%. We may not ever get to quite that way, but there is reason to think that we haven't yet understood all of the genetic contributions of autism spectrum disorders. So in the next 10 or 15 minutes, I'll talk about what are some of the research frontiers of understanding the genetics of autism, trying to fill out the rest of that pie, and trying to then understand, if we can, this tremendous and surprising genetic diversity. And it turns out that it doesn't appear that there's one big chunk of the pie that's been lurking under the surface, but in fact, that filling out the rest of that pie is probably going to look pretty much like filling in the part we've gotten so far. It's going to be one little slice at a time. And there are other genetic contributors to autism that each contribute a few percent or so to the disease. So one thing I wanted to comment on is that we know that there's a very large role for common variants that affect autism risk in certain ways. And so these are variants that we all carry, and that there are probably hundreds of loci across the genome. This is a paper, again, from 2019 that did a GWAS study looking for these common variants that contribute to risk. And as you can see, it identified about a dozen loci, but there's good reason to think that as we study larger and larger samples, we'll probably uncover more and more common loci. But again, these common sites in the genome, each common site only contributes about 1 or 2% of risk to a person. It's just that there are hundreds of such sites. And so these might be particularly important for high functioning kids on the autism spectrum, because we find that the high functioning kids are relatively unlikely when we study them to have these de novo copy number variants or single nucleotide variants. And so it might be that these common risk variants are more important for the high functioning kids. And so, but the way to think of common variants is that they contribute, each site contributes very little to risk individually, but collectively they have a large contribution. But also they have a great deal of risk to autism at the population level. On the other hand, in a given individual, these common sites contribute very little to the risk. And so this explains the paradox where the doctors in the clinic who see patients are very fixated on finding these rare variants, because that's what they see in their clinic. They see a kid that comes in on the autism spectrum, they do a chromosome microarray, they see a 16p11 deletion, and the parents are normal. And so it seems very readily obvious that that's the cause of the autism in that kid. And yet, when you do these population studies, you see that there is a large contribution of these common variants. And the common variants may actually be very important for defining or to contributing whether a given child with a 16p11 deletion manifests as autism or manifests in some other way, like schizophrenia. And there's some evidence, for example, that these common variants might, in some families, rescue some of the IQ of kids who carry one of these deletions. So other sorts of causes that contribute to autism are de novo mutations, but de novo mutations that don't occur in the sperm or the egg, which is how these de novo copy number variants that I've been telling you about come about. The de novo SNVs and the de novo CNVs typically occur in the mother's egg or in the father's sperm, somewhat more commonly in the father than the mother. And so then they are present in all of the cells of the child, but none of the cells of the parent except that one sperm. On the other hand, de novo mutations are happening all the time. Look in your skin and you can see a patchwork of de novo mutations caused by ultraviolet light damage. And so now we're starting to understand that de novo mosaic mutations occur during the development of the embryo. And so in fact, de novo mutations called somatic mosaic, meaning that they're present in some cells of the body, but not all cells of the body, they also, turns out, can contribute to autism. And these Somatic mosaic mutations can occur at any stage during the development of a person. They can occur before gastrulation, in which case they'll be shared by a fraction of cells throughout the body. They can occur after gastrulation, in which case they might be only in the brain and not even in the blood of that same patient, or they can even be in a smaller region of the brain if they occur later in development. So a couple of years ago, Elaine Lim in my lab wanted to know whether these somatic mosaic mutations can be detected when we analyze the whole exome sequence that I've been telling you about, and if so, whether they contribute to the risk of autism or not. And she analyzed almost 6,000 trios and found that, and looked at all of the de novo mutations in the kids in these trios. And that's what's shown in this histogram over here. And so most of the de novo mutations were present on one of the two copies of the genome or the locus at that particular site. And so if you sequence it, that means that the allele fraction is going to be 50%. There's a good copy and there's a bad copy. And the allele fraction is a fraction of those two alleles. And so that's 50-50. But she also found that there was a subset of these de novo mutations that had a much lower allele fraction, only about 0.25%. And these suggested that there are heterozygous mutations, but not in all of the cells of the blood, only in about half of the cells of the blood. And so she overall found that these would be early arising somatic mutations before the separation of the different germ layers. And so that's why they would be detectable in the blood. If they'd occurred later, we wouldn't have been able to find them in the blood. And so work from our group and several other labs published at about the same time show that these mosaic SNV mutations may account for up to about 5% of autism cases. Elaine found that there was an excess of these damaging mosaic missense SNVs particularly in highly conserved exons of brain expressed genes in autism patients compared to controls. Interestingly, she found that patients with mosaic mutations more often had preserved IQ than the patients with germline mutations, suggesting that maybe the patients ended up having the mutation just in part of their brain and not the whole brain. And that seems like it might be a good explanation for how some kids with autism can be so high functioning, almost as if some parts of their brain are working better than others. In fact, Alyssa DeGamma from my lab showed back in 2015 by studying post-mortem autism brains that she could actually see some mosaic mutations that were distributed in some places, but not all in the brain. And we just had a paper that came out a couple of months ago where we actually analyzed the whole genome in postmortem autism brain. And without going into the details of that paper that's published, we also showed a potential role for mosaic mutations that were actually outside of the exome in the non-coding parts of the genome. And important work, as I say, that was approximately simultaneous to ours from 3 or 4 other labs found very similar results. So suggesting that then these mosaic mutations account for another good fraction of the disease. We also published a paper recently to ask whether the copy number variants, which can cause autism when they're in the germline and in all of the cells of the body, whether they might also appear in a mosaic way, and whether that also can contribute to autism spectrum disorders. In order to study that, I worked with our lab worked with Max Sherman, who's a graduate student with Po Rouleau at the Brigham and Women's Hospital. And Po Rou, when he was a fellow, before becoming an independent assistant professor, he developed an algorithm to detect these copy number variants, even when they were only in a tiny fraction of cells. And he'd been using this to study cancer and blood disorders. And we asked them to apply this to looking at autism. And they found that, again, the autistic cases, now shown in yellow, had a significant excess of these mosaic copy number variants present again in some cells, but not all cells, compared to unaffected siblings, controls. And these copy number variants tended to be very large. In fact, on average, they're larger than the copy number variants that cause autism when they're in the germline state. These contribute to a much smaller fraction of autism, less than 1%, less than even 0.5%, probably 1/4 to 1/3 of a percent of cases. So in other words, 1 out of every couple of hundred kids will have one of these mosaic copy number variants. And so the real frontier in trying to understand the genetics of autism is in studying the parts of the genome outside of the exome, the non-coding parts of the genome. And this is an area of active investigation. And really, I think it's too early to have a consensus on exactly the relative contribution of these non-coding mutations. The genome is a big place. The coding parts, the exome, is only about 2% of the genome. So 98% of it was thought of as junk DNA, but we now understand that within all that non-coding part of the genome, there are many, many sites that are on-off switches or regulatory sequences that tell a gene where and when and how to be active. And so it's perfectly plausible that if these are disrupted, they can cause autism as well. But the vastness of the non-coding parts of the genome make this an analytical challenge to try to adduce the non-coding causes of autism. But again, this is a frontier that we're trying to push at the Tan Yang Center, because particularly of the interest and expertise of people like Mike Greenberg and Chin-Fei on the control of the regulation of gene expression in the brain. Now, our own interest in these non-coding mutations in autism actually started by studying families far away from countries of the Middle East where parents share ancestry, because this makes recessive mutations more common and more easy to study. And we've been studying recessive mutations in these populations of Northern Africa and the oil-producing countries of the Middle East actually for about 20 years. And as you can see, cousin marriages are much more common in these areas than others for various cultural reasons. And you can see that the rate of birth defects and intellectual disability fairly precisely tracks that rate of cousin marriage, because of the fact that marrying a cousin does increase the risk of the child having a recessive disease, because the parents share many of the same damaging recessive mutations that are silent in them, but that can cause trouble in a child if they inherit the damaged copy from both parents. And so some former trainees in my lab had collected a couple of hundred autism pedigrees where the parents were related to identify in general recessive autism mutations. And we find when we studied these families that some families had what you would expect. They would have an exonic or a coding mutation that would change the structure of a protein. And this is an example of a large Saudi family where there are 4 affected children with intellectual disability. The parents are cousins once removed, and they have a mutation in a protein called CC2D1A that completely abolishes the expression of that protein. Interestingly, a second family, also from Saudi Arabia, or actually 2 other families, carry actually that same exact mutation in the recessive state, but their kids actually tended to be autistic or have autism with seizures. Again, showing you that even for these recessive mutations, the exact same mutation, in this case, the exact same mutation, can cause different manifestations in different patients that carry the mutation. And this rule holds both for the recessive as well as the dominant mutations. But the other thing that we saw when we were studying these families is that they had copy number variants, all right, but they had 2 different kinds of copy number variants. They had the kind that you expect to see, these de novo heterozygous copy number variants that I've been telling you about, like the ones that involve chromosome 16p. And so we would occasionally find those, and you can see that as this loss of pink signal from this particular segment of the chromosome where the father has dark pink and the affected child has lighter pink for a large segment that might have 30 genes in it. But they would get a second type of copy number variant where the parents were silent carriers and they have that lighter pink, but the parents are fine. And so carrying one deletion of this particular segment of the genome is fine, but then the child has the bad luck to get both of the deletions and has actually absolutely 0 DNA for a segment of the genome. And so we call these copy number CNV zeros, or complete deletions. And so I told you that in America, these de novo copy number variants are about 3 times as common in cases compared to controls. The same actually is more or less something similar is seen in patients from the Middle East if the parents are not closely related. But when the parents are more closely related, in fact, we don't tend to see these de novo copy number variants as being so common. We see these other copy number variants that are very rare in the American population and no more common in cases than controls, but are 3 or 4 times as common in cases compared to controls when you look at parents who have shared ancestry. So these homozygous deletions, or CNV zeros, you can see this suggests again that the majority of these, probably 3/4 of the ones you observe, contribute to autism risk. And we found that you might expect that they would delete an exon, and some of them indeed do, but most of them don't. most of them delete non-coding parts of the genome that contain promoter and enhancer elements. And here's just an example of one. It's a very small deletion here. The nearest gene is over here. It's a gene called UNC5D. UNC5D actually is very important in the development of the cerebral cortex, as shown in the Harvard Neurobiology Department by Gord Fischel. It's very intensely regulated by a transcription factor that turns it off and on. And so you can imagine then how disruption of a transcription factor binding site in the genome might disrupt the expression of that gene. Now, among the non-coding elements, how do we define the non-coding elements that are likely to be important? Well, one way is to look at different species and whether a non-coding part of the genome is very closely identical in many, many different species. Because basically, if something is the same in a mouse and a human, it's very likely it has an essential function, and evolution maintained it as being the same for a very good reason. And so we call these non-coding but highly conserved elements ultra-conserved elements. They're highly conserved across species, and in fact, many of them direct gene expression in the brain. This is a little mouse that's gotten an injection of a blue dye controlled by one of these ultra-conserved elements. And you can see that this particular one generates expression of genes in the brain. There are also they're highly conserved non-coding elements called conserved neuronal enhancers that specifically are active in neurons. And then there's a third kind, which we're particularly interested in, that are highly conserved. These lines, by the way, represent the genomes of different species. For example, mouse, cat, human, and primates. And so there are also sequences that are highly conserved in non-humans, but then are a little different in humans, and we call them human accelerated regions. So the conservation outside of humans suggested that they have essential functions, but then the variation between humans and primates in the sequence suggests that maybe the human function is a little bit different. And so these are the sorts of things that might potentially mediate the evolution of human-specific traits. So that's why we're particularly interested in these as potential mediators of evolution. They're called human accelerated region, or HARS. And Ryan Doan, when he was in my lab before he set up his own independent lab at Boston Children's Hospital, studied these to see whether some of them might contribute to autism risk. Because you can imagine that the social behavior of humans is so different than the social behavior of animals. How do we identify the parts of the genome that might regulate these evolutionary changes in social behavior? Well, we might expect, since autistic kids show such abnormal social behavior, that parts of the genome that are important for the evolution of social behavior might cause autism if they're disrupted. And so, in fact, when Ryan studied these consanguineous families, the cases and controls, he found that, in fact, these HARs were, in fact, significantly more commonly mutated in the recessive way in cases compared to controls, particularly looking at those HARs that also look like they regulate gene transcription in neurons. In fact, overall, you can see that there's about a 5% excess, so that the rate of these mutations is about 7% in controls and about 12% in cases, suggesting they might contribute to actually several percent of disease in these special families that have shared ancestry, where recessive mutations are more common. More recently, in work that's not published, Taewon Shin, a graduate student from my lab, has worked with Ryan to study these now in American populations as well as consanguineous populations. And they confirmed this excess of rare homozygous HAR mutations in HARs. They also show that these ultra-conserved elements show a similar excess in cases compared to controls, showing that these ultra-conserved elements are another source of risk for autism spectrum disorders. And then also studying non-consanguineous trios from places like America and Europe. In fact, they show that there's also a contribution to risk. You can see that the excess in cases is much more modest, suggesting that these may contribute to about 2% or so of cases, which is, again, small, but a significant contribution to the disease more broadly. This just shows a few examples. This is a HAR mutation that's in a distal enhancer for a gene called CUX1. It's a G to A change in a sequence that has the marks that suggest it's an enhancer in the fetal brain. And then that gene, that HAR in the middle of nowhere is connected actually by three-dimensional chromatin structure studies to the promoter of a gene called CUX1. And we see this mutation in 2 different autistic families. And CUX1 is tremendously dosage sensitive. It changes the shape of neurons if you overexpress it or if you underexpress it. This is true in the fly, where loss of the analogous gene called CUT causes the dendrites to be simpler, or if you overexpress it, it causes the dendrites to be more elaborate. And it's expressed in the cerebral cortex in mammals like the mouse and the human. And if you deplete it in mice, it simplifies the dendrites. And if you overexpress it, it causes more elaborate dendrites. And so you can see then how boosting or impairing the level of this gene might have a big effect on the brain. Or here's an unpublished example of a gene that Tay and Ryan have been studying. And it's a gene called IL-1-RAPL1, which is well implicated both in neuronal structure as well as an autism gene. And they see now multiple mutations in one HAR called 3091 that sits inside the gene. So this is the gene IL-1-RAPL1. The little cross hashes are the exons, and then the thinner lines are the non-coding introns. And there's one HAR that actually sits in an intron of the gene, another one that sits nearby, but which is connected to the promoter in terms of the chromatin structure. And you can see that both of these HARs have an excess of these rare mutations in affected autism children compared to unaffected controls. And again, IL-1-RAPL1, like CUX1, is intentionally dosage sensitive. So overexpressing it has been shown in published work, in this case, to simplify the dendrites, and now underexpressing it causes the dendrites to become more elaborate. So these sorts of, this overlap then between non-coding mutations and autism and sequences that may be important for human brain evolution led to the founding of the Allen Discovery Center for Human Brain Evolution that was launched in August 2017, where our lab works with David Reich and Mike Greenberg. And so David analyzes DNA from ancient and archaic samples, old bones dug up in old cemeteries, to try to identify how does our genome differ from that of our ancestors. And then our lab is interested in taking those parts of the genome that are different from our ancestors and figure out which of all of those different varying sequences actually does stuff by studying what happens when they're mutated in humans. Does that cause intellectual disability or autism? And then finally, Mike Greenberg is the world's expert at understanding how genes are regulated in the brain, how we go from the changes in the regulatory region to actually understanding what that's telling us about learning, memory, and cognition. So just then to wrap up and before I stop to take some questions, I hope I've left some time for a few questions. Autism is incredibly heterogeneous in every way, clinically, causally, genetically. A brief summary of our knowledge suggests that de novo copy number variants and larger chromosome rearrangements are likely causative in somewhere between 5 and 7% of kids on the autism spectrum, depending on the particular clinical series. Mosaic copy number variants are causing for about 15 or 20-fold lower rate than that. De novo point mutations we're not exactly sure how the total that they account for. Various estimates run between 5 and 15% of cases. I would say in our clinical experience, it might be closer to 15%. And the mosaic point mutations accounting for a few percent more. Inherited recessive mutations, as I say, easily 3%, perhaps a bit more. And non-coding mutations clearly contributing, but in ways that hopefully over the course of our time at the center, we'll learn more about exactly what that contribution is. The genes definitely implicate synapses. Many different genetic causes can look very similar. We don't have clear ways of distinguishing them when it comes to the behavior. Their common rare contributions are probably additive. Genetics is not the only cause, but it's just useful to study the genetics to try to develop ways to develop better therapies. And as I say, autism mutations can also cause other phenotypes. And so finally, I'll just leave you again with this question: why do genes map so poorly onto specific abnormal behaviors? And again, I don't have an answer for this, but there's, on the one hand, there's undoubtedly a dynamic interaction between inherited common alleles, rare mutations, mosaic mutations. So there's a lot of genetic reasons why that might be the case. But also, I think it's fundamentally that autism genes don't encode behaviors, they encode proteins of the synapse that in turn that allow the ability to learn behaviors. And so they regulate this process of brain plasticity. They regulate the brain wiring that depends in turn on pattern activity and a lot of things that happen after the neurons are formed. I've tried to acknowledge the people from my lab who've done work from my lab that I presented. Just to be clear, much of the work is not from our lab, but is from the published literature. The HAR mutation work by Ryan and Tay, the mosaic autism by Elaine, Rachel, and Yan-Mei Do from Peter Park's lab, who I think I neglected to mention, and we'd be nowhere without our collaboration with Peter Park's lab. Alyssa DeGamma as well. The Mosaic CNV was really mainly done in Po Ru's lab, but we were a collaborator and convinced him to try to undertake that. And Peter Park was also a collaborator on that. And the work on CNV zeros, which was led by Klaus Schmitz-Abe, working with Kyriakos Markianos. Thanks very much for your attention, and I hope I've left some time for questions.
Thank you, Chris. That was a wonderful talk and very clear. Again, thanks for spending the time to tell us about your work. And we'd also like to acknowledge Lisa Yang, who I think is in the audience with us today. Our center would not exist without her generosity and passion for supporting basic research into the neurobiology of autism. So I think with that, Chris, do you have time to take a few questions?
Sure, I'd love to.
Okay, let's see. So the first question is from Annette Aoli. Sorry if I'm pronouncing that wrong. Then do autistic people have a gene for a tendency to produce genetic variants?
Yeah, no, as far as we know, the answer to that is no. When we compare, if I understand your question correctly, and I might not be understanding it correctly, the question is whether autistic kids tend to have more mutations than normal kids. And if I understand that as being your question, they don't. The autistic kids have a rate of mutation overall across the genome compared to their parents that's not distinguishable from normal kids. It's the location of those mutations. Mutations occur randomly throughout the genome, and the lucky ones of us in the audience have those de novo mutations that occurred in junk DNA that has no function. And so it doesn't matter whether the sequence is changed. And the autistic kids have the bad luck that that mutation happened to occur at exactly the wrong spot in exactly the most important gene to make it disabled.
Okay. Another question, our previous work showed that advanced paternal age is highly associated with the white matter aberration seen in individuals with autism. I was wondering if from your work, you were able to identify de novo mutations that are responsible for white matter maturation, myelination, etc. And what are some of those mutations? And do we know if these are common in older father sperms?
Yeah, so I didn't talk about it, but there's a lot of literature that's the same, that's found the same as you, that the older fathers have a higher risk of having de novo mutations in their sperm because sperm continue to be created throughout life, whereas most of the eggs are laid down early. And so advanced paternal age is a modest risk factor for autism, although I don't want to scare people. It's not like it increases the risk from 1% to 20%. It increases the risk from maybe 1% to 2 or 3%. As far as studying disorders of the white matter maturation, I haven't been directly involved in that. Certainly, you're probably familiar with. There are some rare Mendelian disorders that occur as de novo mutations that can cause white matter maturation disorders, things like Alexander's disease and things like that. But as for the very interesting but subtler white matter alterations that sounds like you're describing in kids on the autism spectrum, I don't know whether that is a manifestation of a specific white matter gene or whether that might be a change in the axonal patterns that might relate to some of these more neuronally expressed genes that I was describing in this talk.
Great, thank you. Another question, how can so many different genetic causes produce a similar appearance to autism?
And so I don't have an answer for that. I left that as a question because that is the source of endless mystery and fascination to those. The more you think about autism, the more profound and unanswerable that question is. I mean, it's possible that one way of thinking about it that's not completely satisfactory is that social behavior is the hardest thing that a human being has to learn. We start to walk when we're a year old. We start to talk when we're 2 years old, and we learn things approximately in the sequence of how hard they are, because certainly talking is harder than walking. And that social behavior I still haven't learned it. We said we really don't start to really get socialized until kids are several years old. And you can imagine that social behavior requires tremendous integration, the simultaneous ability to use hearing and sensation and to perceive what someone else is thinking. And so it requires the work of a lot of parts of the brain. And so one possibility is that these genes are disrupting the function of the brain fairly broadly, and that manifests itself first in those things that are the hardest. But I'm fairly sure that that's not the whole answer, that there's more to it than that.
Thank you, Chris. What does ASD probands mean?
Oh, I'm sorry that that wasn't clear from my talk. ASD is an abbreviation for autism spectrum disorders. And so I was frequently using that to refer to autistic children. And a proband is a genetic term referring to an affected individual. It's basically the person that you're trying to understand. This is the reason that you're studying a family, is because you're presented with a proband, an affected child that has a disorder in the clinic, and you're trying to understand the genetic contribution of that.
A question from Clifford Wolf. How does the polygenic risk increase susceptibility to factors like abuse?
Golly, Clifford, that's a real stumper. I don't think I've got a good answer for that. I'm not aware of the, I guess I'm not aware of the literature on the relationship between polygenic risk and susceptibility to abuse specifically. So I'm sorry, I don't really have a good answer for that one.
Okay, another question. Could you give a time by which we could expect more clear picture of genetics of autism and potential gene therapy?
It's, that's, every gene is different. The prospect for, and in fact, even every allele is different. And so the prospect for gene therapy is, the prospect of gene therapy for autism writ large is relatively poor. But the prospect of therapy for some genes that cause autism is extremely good. In fact, we're already doing it in our metabolism clinic. We already see kids with phenylketonuria. We've been seeing them for 50 years. And if those kids were not treated with special diets, they would have autism. And in fact, some are cases of autism from the Middle East that were diagnosed with autism and had typical autism. When we sequence their genome, we find that they had PKU mutations, and that's a completely treatable form of autism. So we're constantly extending our ability. And so it's possible, in fact, that some of the recessive disorders associated with autism are better targets for therapy than some of these dominant ones, because the recessive ones, we're treating a lot of recessive disorders at Boston Children's Hospital. We have more than a dozen various gene therapy trials going on in our division at the very moment. Some of these autism gene mutations that are heterozygous are a little bit more dicey to try to use gene therapy, because the genes seem to be very sensitive to exactly how much of the protein there is, and you don't want too much any more than you want too little. And so those might be a little trickier. On the other hand, there are specific alleles, particular ways in which the gene is disabled. And sometimes those can be attacked. And Tim Yu in our division has been doing some amazing stuff where he's been using antisense oligonucleotides for very specific patients that have very specific alleles. And so I would say that it is an active area of progress, but I don't think that it's fair to promise a sort of a revolution. I think it's going to be somewhat incremental.
Thank you, Chris. And I think the last question here on the list, I think you've already answered with just the previous one. Can genetic information guide treatment for people with autism in any way? And I think that's some of the most exciting work going on right now. A little bit of personalized medicine that identifies the problem for each child and then finds a solution.
Exactly, yeah. Yeah, that's a very exciting area.
Yes. Okay, if there are no other questions, please feel free to ask.
Well, there are, it looks like there's a couple more.
Oh, sorry, I didn't see them, sorry. Okay. All right, another question here. Nothing related to science, but to society. What do you like to say to autism children families?
Well, I again, first of all, I never cease to be impressed and amazed by the dedication and devotion of parents to kids on the autism spectrum. It can be very difficult to take care of such kids that have so much trouble returning their parents' love. And I think that the most important thing is to maintain hope. And that's not, I don't want to give people false hope, but at the same time, behavioral interventions can be very successful for some kids, but also some kids tend to grow out of autism with age. And so it's really, genetics is not, is not a final say. You can read a lot of things online about how kids with a particular genetic mutation might behave, but it frequently does not capture the range of variability. And for kids with developmental disabilities in general, we obviously see kids that are severely disabled on the one hand, but we can see other kids that have very similar sorts of mutations that just do remarkably well.
Okay, so there's a question about whether someone can work in your lab. I'll leave that. That person should contact Dr. Walsh independently, but that's a great advertisement for your lab, Chris. Another question is, why are people with autism prone to schizophrenia?
Right. Well, again, that's a very complex question, and I don't claim to know all of the reasons. I would, as a geneticist, I have a genetics hammer, and so when I see a nail, I think it's genetics. So my guess would be that of this overlapping risk, we know that certain genetic mutations, for example, the 22q11 deletion, creates risk for both autism and for schizophrenia, but the schizophrenia typically does not present until later in life than the autism, which presents earlier. And so my best guess, so certainly some of those cases where people have shared risk for both conditions is likely to be due to the genetic underpinning. I can't say that that's the whole case.
Okay. From Xiaoping Lu, do the genes form functional or regulatory networks?
Yes, again, that's a beautiful question, and that's a whole talk by itself. As I say, the entire regulatory and gene network, all the way from the synapse down to the nucleus and back up again, is just littered. It's just highlighted by autism mutations all up and down the way. So there certainly are actually multiple independent functional and regulatory networks. For example, there's a lot of genes involved in chromatin biology. And multiple chromatin protein complexes where several of the subunits carry mutations in autism, or there are protein complexes in the synaptic spine where multiple proteins can be mutated in autism and so on.
Okay, great. And from Heather Hazlett, wonderful talk. Thank you, Dr. Walsh. And actually a comment in the chat from Nigel McClancy. Any comment on the apparent increase in ASD diagnosis rates?
Yeah, so very important question. Very complicated answer. We're getting, part of it is we're getting better at diagnosing it. At least one epidemiological study looked at the rates of autism in various counties to try to identify risk factors in the environment that might predispose to autism. And what they found was the strongest predictor of autism diagnosis rate was the closeness of an autism diagnostic center. And so to a certain extent, our ability we have more psychologists and we have better tools to diagnose it than we used to, and less stigma. And so I think a certain amount of it is knowledge. There's also a lot of talk about diagnostic substitution, that some kids that in a previous generation may have been diagnosed with intellectual disability are now diagnosed on the autism spectrum. In some states, there are better insurance reimbursement for treatment of autistic kids than for intellectually disabled kids, because the autistic kids are considered to be more hopeful. And so there might be an element of that. There might be a contribution of the fact that people are having kids older in life, because I did mention that older parental age increases the risk for autism, but I do want to emphasize that that's a modest increase. And so that's not likely to account for a lot of it. So I think a lot of it might be ascertainment, and some of it is probably diagnostic substitution. And there may be other things that are not so clear from the epidemiology.
Thanks, Chris. And another question, could you talk a little bit about the genetic mutations related to sex differences in ASD children?
I'm not exactly sure what the question means, but it may be relating to the fact that I somehow managed to gloss over that autism is 5 times as common in boys than in girls. So that's a very, very important part of autism that just didn't come up in my particular slides. Exactly why that is not totally clear. As we understand the genetics, we can see that there is a protective effect of being a girl, so that girls that carry the same sorts of genetic mutations as a boy with autism are more likely to be asymptomatic, are more likely to be cognitively intact than a boy with the exact same mutation, as though the male brain is more fragile. The reason for that, the genetic mechanism for that, is really fairly profoundly mysterious at this point. We really don't know, and then that would be obviously a very important thing to find out. If we could figure out the basis of the female protective effect, maybe we could do whatever it is that females are able to do better than boys to protect themselves from autism in such a way that might improve the outcome of boys with autism. But that's a real frontier at the moment.
Yes. Okay. Another one. Could you please briefly review your point about autism genes encoding proteins, not behaviors, and how this helps explain why genes map so poorly onto specific abnormal behaviors?
Right. Well, I guess I don't think that the fact that autism genes encode proteins and not behaviors, I don't think that that's necessarily an adequate explanation for why genes map so poorly onto abnormal behaviors. But I guess what I'm saying is that the brain is just a very complex and beautiful thing, that it has this inherent plasticity, that it's constantly changing, and the work that Chinfei does just shows that so beautifully. That the synapses are hopefully everyone will leave the lecture today with synapses a little bit different than the way they came in, because that is the basis of everything that we learn. And so our brain is not hardwired to have particular behaviors, but instead it's endowed with the ability to learn behaviors. It's endowed with a certain early sort of rough mapping, but then ability to be constantly changing that in response to what happens. So basically there's just a long road between the genome and our brain, and that the relationship of them is somewhat indirect. That's all I'm trying to say.
Thanks. Very beautifully said. Anyone else? Well, if there are no other questions, I want to again thank Chris for a wonderful talk, and also a great question and answer session. With that, I think we'll complete our webinar. And till next time.
Thanks, everybody. Bye-bye.
Take care. Bye-bye.
Bye.